So good evening. May I request now Dr. Himanshu Davda, the principal of the college, to formally extend a very warm welcome to a very dear friend, a very good researcher, and as a human being, an excellent human being, Dr. Bimalendu Nath sir. We remember all your excellent lectures that you have delivered in our college. And we thank you once again for accepting our invitation. Now I request Dr. Himanshu Davda to extend a formal welcome to you, sir.
Thank you very much, madam. Very good afternoon, everybody. On behalf of Hindi Vidya Prachar Samiti's, right?
Yeah, we all
on behalf of Hindi Vidya, so you can hear me?
Yes, we can.
Yeah, okay. So on behalf of Ramnarajan Jhunjhunwala College, also on behalf of members of Association of Teachers of Biological Sciences, on behalf of our mentor, Dr. S.M. Karmakar, who is there in the room, and also on my personal behalf, I extend a very warm welcome to our guest speaker, Nath sir, today for this guest lecture series being organized by DBT Star College Department of Biological Sciences of RJ College in association with ATBS. Indeed, today's lecture is a long-awaited lecture because listening to Nath sir is an opportunity by itself. The manner in which sir develops any topic during the lecture is a true treat for someone who understands what science is and also believes in appreciating that science is something which should be handled very gently, sympathetically, and yet courageously. Another very beautiful aspect of Sir's personality, which I would like to share here, is that Sir believes in appreciating work of others as well. This is the 11th lecture in the guest lecture series, and this is the 11th time that Sir is present here. A true scientific temperament calls for a lot of inclusiveness, a lot of open-mindedness, and acceptance of every little idea that comes from any of the source. That's what our Nath sir truly believes in. Sir, we are truly honored to have you here today. Thanks once again for accepting our invitation for this talk, which we all are, as I said in the beginning, eagerly awaiting for. May I now request Kale sir to give a formal introduction of Nath sir, and then we begin with the lecture. Thank you, everybody.
Namaskar and welcome to all. Well, it's a pleasure to briefly introduce Professor Bimalendu Nath. He has been my friend and colleague in Association of Teachers in Biological Sciences. I'm happy that we had talks from the president and now the second vice president as well of Association of Teachers in Biological Sciences. Well, Professor Nath is currently an emeritus professor at the Department of Zoology, Savitribai Phule, Pune University. He teaches genetics and allied subjects to UG and PG students. Well, his areas of interest have been stress biology and chromosomal genetics. Professor Nath has graduated from Guwahati University, Assam, and did his master's in Life Sciences Department from Vishwa Bharati Central University, Shantiniketan, West Bengal. He did his PhD from Banaras Hindu University. He has worked at Malaria Research Center, National Institute of Communicable Diseases, Delhi, as a research trainee. He did his postdoctoral work and was a postdoctoral associate at the Molecular Biology Unit of Institute of Medical Sciences, BHU, and joined University of Pune in 1992. Over years, he has established a stress biology research group using Drosophila and Chironomus as model organisms. He is a member of the Society of Experimental Biology, United Kingdom, and Cell Stress Society International of USA. He is a member of the editorial board of the Journal of Radiation and Cancer Research. Currently, he is contributing as a visiting faculty at different life sciences departments of Savitribai Phule Pune University campus. He is a recipient of Joag Award for Best Teachers in 2014 from University of Pune. Professor Nath has published many research articles and also chapters and books on his chosen model systems. His reviews on these model systems have appeared in national and international peer-reviewed research journals. He is active in writing popular science articles for UG/PG students as well as teachers in biological sciences. He has served as Vice President of Indian Society of Cell Biology, and as I said earlier, he is currently the Vice President of Association of Teachers in Biological Sciences. For this association, he is also Editor-in-Chief of the online magazine, or rather periodical, I should say, of ATBS. He has a number of ongoing national and international collaborations, has worked as visiting scientist at I.M.T, Philipps University, Marburg, and Freie University, Berlin, Germany. He has delivered lectures in Germany, Italy, China, Norway, and Czech Republic, as well as Spain. He has been a leader of Indian team at the International Biology Olympiads and actively participating in the selection as well as training camps of the team members to go to International Biology Olympiads. So, sir, we all are eager to hear you. And the title of your presentation is Researching to Learn and Learning to Research with Drosophila. Over to Bimalendu, sir.
Good afternoon, and thank you, Dr. Kale, Dr. Usha Madam, and all others, my teacher colleagues. To ATBS for having me here. I am greatly honored. So without taking much time, I just start the talk which I have prepared on, mostly targeting the undergraduate students and the young teachers who are going to start their research mostly in colleges. So the content of my talk, the way I have structured, it will have a historical account in the beginning, salient features of Drosophila as model system. I'll talk about several Nobel Prizes and a few research areas using Drosophila as model system, and how Drosophila is used for translational research. Drosophila, how it is can be used to address environmental issues, and a few published works from my lab, and then the future directions and the concluding remarks. So when you talk about model organisms, a model organism is a species that has been widely studied. usually because it is easy to maintain or breed in a laboratory setting and has particular experimental advantage to address specific scientific questions. Now, a model organism for genetics may not be a good model organism for developmental biology or neurobiology and so on. Drosophila melanogaster, which will be the main focus today, and you can see many other model organisms used in genetics like Saccharomyces cerevisiae, the simplest eukaryotic model system, single cell model system, zebrafish, then Caenorhabditis, Mus muscula, the mouse, and in the Arabidopsis you can see on the left panel. So these are the model organisms which dominate the genetic research worldwide. Now, Drosophila melanogaster, which is commonly called fruit fly, is one of the most well understood amongst all these model organisms. Now, since the first decade of the last century, in 1900, when the Mendelian principles were rediscovered, until the present day, Drosophila melanogaster has been central to many, many breakthroughs in genetics. And now Drosophila is used as a model organism to study disciplines ranging from fundamental genetics to applied genetics, epigenetics, to the development of tissues and organs, and also in biomedical sciences. So I'll try to give you a bird's eye view, the contemporary profile of this model organism in the global scenario. Now, if we look at the natural history of Drosophila, it originated somewhere in the sub-Saharan terrain of Africa, and then it started spreading through human activity and mostly transport and the cargo, and now it has become cosmopolitan. There are hundreds of species of Drosophila, and now you will find all over the world in different type of habitats, and mostly found near the compost, rotting fruits and vegetables. Now the question is, why this fly? So you can see this the picture of the scientist Thomas and Morgan, who is generally called the father of Drosophila genetics, who first introduced in the first half of the last century, first decade of the last century. And actually everybody was trying to find out a suitable animal model system where the principle of genetics can be tested or validated. And he was pioneer to introduce Drosophila and for many path-breaking basic research. And it is sometimes often said that Drosophila helps you to understand the grammar of genetics. Okay, you learn the language of genetics, alphabet of genetics using Drosophila. So what are the advantages of Drosophila for genetics and translational research? No ethical problems, no restrictions according to animal protection laws. So one can overcome some of the hurdles, legal hurdles and ethical hurdles, easy and cheap to maintain in large quantities, time and cost effective handling. Genetic manipulation is fast and inexpensive, and availability of plenty of resources. Like in the Vienna, you have the genome-wide RNA library. There are many stock centers like Bloomington Stock Center in USA. Then in India, you have the government-funded stock center in Mysore. So one of the advantage of being chosen as a suitable model for genetics is the short generation time. So within 10 days, you go to the next generation, and is easy to screen. The genome is fully sequenced now. In 2000 AD, soon after the human genome went to the sequence, went to the public domain, the Drosophila genome has been fully sequenced and it's appeared in the public domain. Basic signaling pathways and cellular processes are well conserved. And so the question which you try to find out in mammals, you try to explore in a simpler model, then Drosophila can be a good model system. Low redundancy, reduced number of paralogous genes compared to vertebrates, non-pathogenic. It is safe to handle this in a classroom, laboratories, and research. And there are balanced chromosomes which allow to maintain the lethal mutations in particular. So now, history of fly research. Now, it all started, as I have mentioned, that from Morgan's lab. And so just give me a minute. So the discovery of the white-eyed fly changed the whole scenario, and he discovered the sex-linked gene, and which and also many other genes which doesn't follow the typical Mendelian pattern of inheritance. And subsequently, he and his group, like Alfred Sturtevant, Calvin Bridges, Muller, and they have contributed a lot. And the first, the linkage map, recombination map was discovered in Drosophila. Now, 6 Nobel Prizes awarded in the category of physiology of medicine to 10 scientists for groundbreaking work in Drosophila, and started from Morgan, and he's one-time student, and the Muller, and so on. So historically, the first Nobel Prize you see in the Drosophila is the Thomas Morgan for the role played by the chromosomes in heredity. Then 1946, sometime, when the aftermath of the World War, the X-ray or different type of radiation was utilized for peaceful purpose, and their effect as a mutation. And one of the the seminal work which was done by Muller, based on a Nobel Prize. 1995, Lewis, Naslin, Pollard, and Wiskhaus, they have they discovered the genetic control of the early embryonic development, and I'll talk a little bit about this. And in 2004, although mostly the work was done in the rodent or the mouse model system, but the Drosophila was used to validate the result, and the autoreceptors and organization of the olfactory system was discovered with Axel. The single Nobel Prize was given in that year in the physiology or medicine category. In 2011, Jules Hoffman for activation of innate immunity, and so on. And and 2017, the Hall, Rosbeck, and Young, they got the Nobel Prize for discovering the circadian rhythm. So this 1995, the Nobel Prize went to Lewis, who worked, in the late 30s, and Naslin, Pollard, and Wiskhaus. They discovered the genes that regulate embryonic development, and it has allowed us to comprehend how a single cell develops into a complex multicellular organism. You can see the fruit fly is the normal fruit fly. It has a halter and the second pair of wing, which is very reduced to use for balancing during the flight, is called halter. Now, due to the developmental misregulation, you get a mutation, and you get a wing developed from the halter. So this is basically Drosophila has a single pair of functional wing, and you have the four-winged fly. So this type of developmental anomalies was discovered by this scientist. And also you can see the leg is coming in the place of the antenna. So this type of misregulation was discovered. And also later on, the following decades, it was found that the human also has the same type of the genetic control of early embryonic development. And so the discovery which was made early into the 30s by Lewis, and later on by Naslin, Pollard, Wiskhaus, and many other scientists, it has translated into the human developmental genetics paradigm. This is actually an excellent work by Gehring lab in Switzerland. Actually, everybody thought that he also will be nominated for Nobel Prize along with Lewis, Naslin, Pollard, and Wiskhaus. However, his lab discovered that how ectopic expression, that is in the expression which is wrongly placed in the body of a homeotic gene, ILS. ILS is a gene causes the formation of eye structure in the leg. So eye is being formed in the leg where it should not be. So this type of misregulation was discovered by Gehring, and our understanding of the genetic control of development and how a single cell get differentiated and develops into a complex multicellular organism, to shape. And next, there's this path-breaking discovery, by Hoffman in 2011. By the way, Wiskhaus visited visited Pune last year. He came to ISER and also gave a public lecture in IUCA. And Hoffman came around 2012 or 13, I don't remember, in Pune, gave a lecture in NCCS. So he discovered innate immunity using Drosophila as a model system. Then 2017, the three scientists, mainly, the Jeffrey Hall. I'll mention that he started his work long back in early 60s or 70s, and he's the same lineage of Samuel Benger, who discovered like R2 locus, and then shifted to behavioral genetics. So in Brandeis University, USA, he started his group, and they discovered many the genes which are responsible for behavioral control of the fly, and making us believe that the genes control many of the innate behavioral patterns. So the gene which was discovered by this group, the period genes which control the circadian rhythm, that is the rhythm which is a daily 24-hour cycle of day and night, the resting phase and the activity phase. So they found that the period gene that controls, and if the period gene is mutated, then the Drosophila has a kind of aberrant the behavior which is non-circadian. And this circadian rhythm has been observed in the living organism for centuries. We know about circadian rhythm for many, many decades, but we never understood the circadian system has been, is controlled by genes too. And now we know that the human has the same similar type of genetic mechanism. So the homologue of the period gene of Drosophila, you can see the one or two papers I'm just showing, that the human period gene HPER it is also found in the human. There are other genes, so this subject is called chronobiology. So genetics of chronobiology is developing very fast, and Drosophila is one of the prototype model system. And many of the diseases are linked to this circadian misregulation. And period gene has been implicated for many of these chronobiology related human diseases. And there are lots of the hope that many other rhythm related disorders will be solved soon. So this is one example from human. So basically what we know from Drosophila, and by the way, which is beyond the scope of today's talk, that those who are interested can go to the internet and just give a keyword called sleep and Drosophila. You will find number of research, a very interesting piece of information that how there are biochemical and genetic pathway which controls the different type of sleep and wake rhythm. So this is one example when the period gene is misregulated. There's a mutation, so you get a disorder, sleep disorder, which is called familial advanced sleep phase syndrome. So these are the contribution of Drosophila if you when it is translated to human scenario. So there's some more example that Drosophila to convince you that Drosophila is emerging as a chosen model for molecular neurogenetics. Okay, so not only sleep, the memory, the learning, these are very complex mental faculty, and Drosophila is being successfully used to genetically or biochemically dissect the cascade of reactions that occur when we retrieve any experience which we call the memory or the learning. So there are many learning mutations and the memory deficient Drosophila flies. So you can read many of the interesting papers, and you can write to me for full text, and I'll be happy to share with you that there are small training camps are there which you can where you can train the Drosophila. So short-term memory of Drosophila is for 20 minutes, and they forget. So there are flies which forget every minute. So if you train them, they forget. It's a conditioned, like the famous example of Pavlov's the dog, the condition and reflex. So Drosophila, you can condition them and also find out their memory span and so on. So these are the neuronal control of Drosophila. By the way, Drosophila has 10⁵ neurons in their cerebral ganglia, and these 10⁵ neurons can control all these behavior, so complex behavior. So starting from the learning and memory, feeding, flying, walking, the sexual like behavior like courtship, circadian rhythm, sleep, aggression, grooming, drinking. So I can talk for hours on each one of them, but I'll give some of the interesting features to generate interest amongst you. So there are some mutants in Drosophila which are defective in learning and memory. These are called dance mutation, amnesiac mutation, cabbage, and rutabaga. And basically, it all starts from the G protein, and then there is adenylyl cyclase mediated like pathway, signal transduction pathway, and ultimately protein kinase C. There are lots of calcium, the flags. And so each one has a definite, role in this pathway. So you just perturb one of the pathway, and then you get a mutation. So basically, learning and learning and memory is a complex faculty, and it has different the facets. So each step can be handled by different sets of the genes and proteins and neurotransmitters. There are excellent studies are there. So this is a very hot topic now. So there are other mutants like sitter and rover. These mutations in the behavior. Even there are Drosophila, like sitter. There are food in front of the Drosophila. It's a hungry because you have kept the fly under starving condition. But even then, it's so slow to go and have the food because there's some genetically it is defective to sense the food around. And there are voracious feeder kind of rover. So this type of gain of mutation, then loss of mutations, so all type of mutations there. And as I mentioned, the period genes, there are other genes also associated like timeless, double time. So it maintains a molecular clock, like in the kind of a feedback control that like there is a transcription and then the translation. There is a very nice regulation, and that is controlled by the day and night cycle of the Drosophila. So I just want to show you, there's a very interesting paper you can read. This is freely downloadable. You just, they go to this PNS paper, and full text is available. You can see this aggression of this Drosophila flies. That they are, so there are, on the left side, the figure 1 of this paper, it has shown different type of, agonistic behavior or aggressive behavior. Okay, male fly, fight, then female aggressions. And many times this is not context dependent. Like, there may not be any the shortage of food, but even then they're not hungry. But even then, some individuals are very aggressive. When they move, they just hit the other one. So those individuals were isolated. Their breed, as per the protocol of the true breeding of the Mendelian fashions, and lots of interesting discoveries are coming up now, and maybe we'll very soon we'll get a clue of many of the aggressive behavior found in higher organisms, including human. Okay, this is another from the supplement of the same paper of the same group. And you will find that the true males are fighting, and not only the other one, the other one is like grounded, but even then it doesn't leave the other individual. Okay, so Drosophila has emerged as a tractable model to study neurogenetics, and you can see there's the whole the cerebral, the ganglia, which is called mushroom body. It has been fully mapped, and then we now know exactly which part of the cerebral ganglia is fired for some kind of stimulus. So the stimulus response and the whole brain has been mapped in Drosophila, and it is now people are now trying to find out which mutation cause the lesions in which pathway and so on. So now I just shift gear now and come to the current scenario of Drosophila in translational research. So Drosophila is a model for translational research in 21st century. So let's begin with what is translational research. Now, popularly it is called B2B, or laboratory bench to the bedside. So it transfers knowledge from basic research to the clinical research. Now it has been extended. The translational research now aims to make findings from basic science useful for practical application, not only for human health, environmental and agricultural sciences. So this is basically the translating the basic research finding to the applied research, where it can be exploited commercially for the benefit of the human being, agricultural areas, and so on. So now this is a path-breaking, news for the Drosophila scientist. So in 2000, some of you may remember, it was President Clinton in USA. He announced the discovery, the human genome was fully sequenced in 2000, and within 6 months, the Drosophila genome was also fully sequenced. In the next 5 years, the bioinformaticists, when they compared the sequence of the human and the Drosophila, it was found the 75% of the human disease-causing genes are, they have found that they have homologs in the fly. So flies develop a fast and are highly amenable to genetics and other experimentation, ideal to study genes, biological process, and even a mechanism of diseases. And you can see there are lots of texts, lots of books here, and then I have just picked up only 3 just to impress upon you that this the reference books, the textbooks, they tells you about the use of Drosophila to addressing many questions in human diseases. Okay, 20 years ago, the trends in genetics, one of the very well cited in the review, it tells you the Drosophila in cancer research. Okay, and so diseases in human are often very similar to those that can be studied in Drosophila. And it has been estimated that, as I just mentioned, the 75% of the known human disease genes have recognizable match in the genome of fruit flies. Therefore, the Drosophila flies are often used as test tubes to pioneer research on a gene or a biological process of interest, followed by work aiming to determine whether the new understanding obtained in Drosophila is applicable in mice or even in humans. And that's what the term translational research means. So this is they published long back, and to show a kind of correspondence of the fly genes and the mammalian genes product, which are implicated in cancer. And so the large repertoire of genes in the fly, which has like counterparts in mammalian genes, which has been implicated for cancer. So of course, somebody they can argue that okay, there is no the progression of the cancer or tumorigenesis cannot be compared in human with the fly. It's true. There is no blood, there is no angiogenesis on, but the initial, the mechanism of the onset of the tumorigenesis has been found. The triggering of the genes, which are which are implicated as oncogenes, it is this kind of evolutionary conserved. And Drosophila, there are many cancer models. So Drosophila, you can induce cancer in Drosophila, and many of the drugs can be shortlisted to find out their efficacy in the fly. So many pilot studies of the cancer drugs can be done in flies, and then one can try that in the cancer cell line and to address its efficacy in human. So now you can see many blog sites, many the groups who call them homophila. So basically, it's a Homo sapien, that is a human, and Drosophila, there's a Drosophila. So this group, they just, they translate Drosophila research to the human and the vice versa. So this is a new emerging, the group of, the scientists who translates findings in Drosophila to human. That's one, it is a current trend now. So you can read the recent articles in current opinions in genetics, current opinions in development biology, and current biology journal. You can find the once in 2 months and 3 months. There are many the paper appearing, and this justifying the importance of Drosophila in human genetics. There's some of the visually delightful, the pictures I'm sharing, that these are the transgenic Drosophila, where it's expressing the green fluorescent proteins. Okay, so there's no time to tell you the protocols and so on. Like there's a transposon tagging, there's a P element which is found in Drosophila. So you tag, and you can sometimes do enhancer trap, and you can express the green fluorescent protein. Okay, there is a blog site where you can read in a homophila that, okay, if you have hundreds of Drosophila bottles with the transgenic bottles with expressing this green fluorescent protein, so in a room you do not need an external light, and it's quite well lit. So these are some of the beautiful images. So this to impress upon you some of the recent the articles you can read, and like fruit flies in the biomedical research, Drosophila as a model organism in neuropsychopharmacological research. This is in 2016, and this is 2018. This is the Drosophila models for human disease, and a beautiful review. And so basically, flies can be a very efficient starting point of a translational pipeline. So you can just start with the random mutagenesis in many flies, and you can use the P element mediated mutagenesis. You can use the EMS mediated chemical mutagenesis, or you can use the radiation mediated, like gamma radiation, to mutagenize randomly, and select the phenotype which is of your interest, identify the responsible gene, and then you can study the gene properties and function. Then it comes the combination of the computational, the softwares, bioinformatics, and many other tools. So here it becomes interdisciplinary, identify, study, and related genes in human, and that there you can translate the Drosophila findings in human. Now, Drosophila, to address some of the environmental issues. I'll request Dr. Kale, sir, to tell me when to stop. I think I have another 20 minutes or so. Yeah. So
yes,
so
you can continue, sir. No problem.
yeah. So you can just give me a signal.
Yeah.
So Drosophila, to use environmental issues, and new jargons and new terminologies are coming up now, like so which I'm coming to that very soon. So Drosophila and climate change research. All of you know the global warming, climate change. These are the pressing issues, and there's a global concern. And so you can see the fruit flies adaptation to climate change by metabolic rewiring. In 2018, there is a the sensational news from a group. This you can read there. These are the news items. There you can see climate change, and there is a genetic flux in the natural population of Drosophila robusta. So then you can, this was studied in USA, the natural population, and the how fly populations are quickly responding. So, and then this tells you about the plasticity, about the ability to adapt, and the genes which are important, or the physiological mechanisms which may be important because Drosophila cannot migrate to a safer zone to an extent that it cannot it cannot fly more than 1 kilometer, and so unless it is passively they're taken away by the wind and so on, or human cargo. But it gives you a clue that how an organism can adapt to this type of environmental stress. So I just want to spend next 5 minutes or so, or 10 minutes to show you that how polyethylene chromosomes can be used as an exploratory approach to address some of the environmental issues, which is called toxicology. So now this is mainly for the students. Some of the slides that I have prepared. This is a cell cycle. This particular slide, the figure I'm very fond of, because every book they gives a cell cycle, a kind of a picture where the chromosomes are not shown. They just show cells or something else. This is from a very old textbook by D. Robertis, where since I started my career in the chromosomal genetics and the cytogenetics. So I love this picture, and wherever the opportunity comes, I have a habit of putting it in some context or the other. So you can see here in the S phase that the chromatin material is loose, is decondensed, and gradually it starts condensing, and the in the M phase it is highly condensed, and after that there's an NA phase, and then again it becomes a decondensation. So basically condensation, decondensation, it goes cyclically in the cell cycle. If you look at the behavior of the chromosome, you can see this type of condensation and decondensation of the chromosome. I'm just bringing your attention or drawing your attention to the chromatin, the chromosomes. And now there are the tissues. There are many organs in the lower animal groups, including diptera and insect and Drosophila, where it can be studied very efficiently. So where the polyethylene chromosomes occur, they do not these cells, they do not undergo M phase. So basically the cell cycle, the M phase is missing. So it is the S and the G, and this is called endomitosis. So what happens during the S phase? The chromatin materials replicate continuously. So it doesn't stop after one division, and so it continues. It continues, and this resultant chromatids remains tightly associated, aligned laterally, and giving rise to a multi-threaded cable-like structure, and which is called polytene. Poly means many, tene means thread, and it's sometimes called giant chromosome. So here you can see this the schematics on the top. If you can see my cursor, you can see that it's the chromatids are continuously they are getting replicated without getting separated, and all the daughter chromatids they remain side by side, and so in the Drosophila that give rise to a polyethylene chromosomes and so on. So the Veikko Sorsa, she is the first lady from Finland who had in late 50s and 60s, when there are electron microscopy didn't advance much. She developed a protocol to they show the fixation and then the separation of the polyethylene chromosome, and under the electron microscope you can see how many chromatids the chromatid threads are. There is a beautiful picture generated by Sorsa. I picked up from her book on polyethylene chromosome and electron micrography in 1988. This is out of print, and then this book didn't attract much today's electron microscopy, but one should read this book to know how one can innovate ways in difficult times when the facilities are not available. Anyway, so the giant chromosomes of polyethylene chromosome, you get the 200 times sort of an amplification of the size if you compare the mitotic metaphase chromosomes. Okay, this is another very interesting picture I picked up from World of Cell by Baker's 9th edition. On the right side, the picture, the right panel, you can see the high voltage electron microscopy, like a polyethylene chromosome from Drosophila, and you can operate at 1 million volts, and you can see, you can really literally count the number of chromatids which makes the bundle like structure and polyethylene chromosome. So now let's come to the original issue, that how polyethylene chromosome can be used for environmental studies and toxicological studies. So before that, I just want to tell you something, that a synapsis, this is a term all of us we know, that it is basically we use it for meiotic chromosome, where the paternal and maternal chromosome, they come together and then the synapse, and then the crossing over takes place. And this is a phenomena which you find in the germline cell, and it is in the prophase of the meiosis only. So in the body cells, the somatic cells, synapsis doesn't occur, because synapsis, this step is not there in the mitotic divisions. But as I have told you, the polyethylene chromosome, polyethylene cells, in their cell cycle, you recall now a few slides, previous slides, I have shown you there is no division. So basically, after S phase, they continuously chromatids divide and they remain together. And what happens? The paternal and maternal chromosome, they come together and remain in the synapsed condition in somatic cells. So in polyethylene chromosome, paternal and maternal chromosomes, okay, cannot be distinguished. They just remain tightly synapsed. Now, please remember this point to understand the successive slides which I'll be showing. Okay, so this is a terminology. It's very amazing, maybe for many of you, but this is a kind of a trendy, the term which is now used in the Western countries now. Yes, I am a drosophil-toxicologist. I use Drosophila for toxicological studies because EPA, the Environmental Protection Agency, which is one of the leading the environmental protection agency in USA, based in USA, they have recommended Drosophila to use for toxicological assessment studies, and there are recommended protocol like WHO. EPA is also they publishes the recommended protocols, and then one can test many of the drugs or compounds which are to assess its toxicity. So this is a Drosophila toxicology, how you can do using a polyethylene chromosome. Okay, so that's what and then to understand this somatic synapses, which I just mentioned, that paternal and maternal chromosome remain associated tightly the synapse. And so how one can use a Drosophila toxicology. So if one member of a homologous pair is altered by deficiency, duplication, inversion, or translocation, an irregularity occurs in pairing. Okay, I'll just give an example. Like I'm wearing the shirt, and suppose there is while putting the buttons in the respective the holes, they misjudged. Then when I wear the shirts, you can see that there are the button has gone to the wrong place, and so some part of the shirt, piece of the shirt is like the looped, or like they showing a kind of a aberrant sort of visually it is evident. So this type of things, let us just see how it occurs. So this is a classic the schematic the animated picture I'm showing, the deletion. Okay, deletion occurs in it's a terminal deletion, like you see a human the chromosomal disorder called Cry-Duchet syndrome, where the small arm of the chromosome 5 you get a deletion. There are retinoblastoma where you have the deletion of the chromosome 14. So there are many examples in human scenario. Now somatic synapses, as here the schematically you can see, this is if you look at the just 5 micron the span of the polyethylene chromosome, and you can see the paternal and maternal the part is how is it tightly intertwined. Okay, now look at my follow my cursor, which I'm just showing here. So here, if some genes or the part of the chromosome get deleted, so there will be problem of the corresponding part of the maternal or paternal to match. So if one segment is deleted, question is how the synapse chromosome will look like. And let's look at it. So let's just follow this animated, the picture. One of my student has they made this animation, and hope you will enjoy this animation. So let's say schematically, these are the genes A, B, C, D, E, F, G. Okay, so deployed. They have they have the double copy, and also this is the female. By the way, those who are not familiar with the Drosophila, if you look at the black tipped abdomen, this is male. So this is male, and if you do not see this a black tipped abdomen, so this is a female. So this is a male and the female Drosophila. So this is the father and mother for the future generation, and it looks like the chromosomes are perfectly in, or the genes are perfectly in order, and when the spermatogenesis and oogenesis, meiosis occurs, and this is the baby fly in the F1 generation, and the paternal the haploid part chromosome, the one chromosome from mother comes that constitute the F1 progenies, the chromosome constitution. Now here it's a perfect synopsis. You can get A to A, B, B, C, C, D, D, E, F, F, G, G. So synapses will occur. So there is a perfect correspondence. Okay. Now let's say you challenge with a cytotoxin, a hypothetical compound. So the male, they now is bombarded with exposed to the a compound which has a cytotoxic effect, and the part of the genetic material is deleted from the chromosome. So now you see the A, B, C, D, E is missing. So now if the father with a defective chromosome, okay, structurally aberrant chromosome, and the mother with a perfect chromosome constitution, they contribute, and in the F1, the baby fly. Okay. Now you see, now you have a problem. So you have from the mother side, you have the full, complements A, B, C, D, E, F, G, but the paternal chromosome has something missing. Now the question is, in polyploid nuclei, how will they synapse? Okay. So how will they synapse now? So there will be a geometrical constraint. So they can synapse by with a buckle. So A and A, B and B, so they will join, and F, F, G, G, they will join with the constant, and C, D will remain out in one of the homologue. So synapses will be defective. So there cannot be 100% synapses. And so this is a picture from a textbook, a Gardner's picture. So you will see that the one of the homologue where you do not see the synapse is buckled. So that means in the other half of the chromosome, here it is 3C1, 3C up to the 3C12, 1 to 12, these bands are missing. So basically there is a deletion. So now if you see the deletion, you go back and find out that the father's chromosome, or it can be the mother's chromosome, somewhere the deletion has occurred. So the question is that if the father or mother, any one of them, has been exposed to a toxin compound, and so you can confirm that, okay, it can cause deletion at a particular concentration. So this is with the polyploid chromosome when the synapsis is lost, is discontinuous. So this type of buckle formation, you can see this is some of the pictures from my laboratory. You can see the buckle here. You can see the buckling effect, and then you can so you can conclude that yes, synapsis is not complete. So let's look at the other type of chromosomal inversions, aberrations, like inversions. So inversions can be pericentric. That is, it can include the centromere, and then there is a 2 breaks, and then there is a rotation, and then you can have a totally inverted sequence. Okay, it is quite rampant in the animal kingdom, plant kingdom, and inversion can occur due to environmental, the toxicity or exposure to radiation and so on. And this is called paracentric inversion, where the breakpoints are away from the centromere. Okay, so both of the inversions. So let's see now, go back to the same type of, the animated model. Now here again, the male fly is like, this is, okay, this is exposed. And you see, if you look carefully, due to this, there is a inversion here. So instead of ABCDE, here it is ABEDC. And the mother is not exposed and nothing has happened. Now, if the father is contributing such a chromosome where there is then inversion, so what happens? So F1 generation. So father has contributed ABEDCFG. A part of the chromosome is inverted. Mother has contributed the chromosome, which is perfectly matched. And now you see, now they can synapse forming a loop. The earlier one, the deletion case, you have seen the buckle, and here you can see there is a loop. So you can see such type of loops in the real picture. Like, this is one of the research project where I was, working on the screening of some of the industrial products, and we have found such type of, the inversions. There's an article which, the earlier I've written 20 years ago in Resonance. It's you can download the full text, and you can read the details about all this. So these are some of the the pictures to show where the synapses is not totally complete. You can see paternal and maternal homologs got separated. That means there are no matches here. Okay, so that the corresponding the genes on the other half is either there is a deletion, or there are inverted, or there are multiple type of the aberrations. Okay, this is as an entire telomeric region has no match. So this is called synapses. So this type of synapses, loops and buckles, gives you an indication that the compound to which the fly has been exposed have toxicity potential. So this, again, another cytological parameter like the puffing, which is a cytological marker of the environmental stress. And the puffs are the active sites, transcriptional active sites on the polyethylene chromosome. And basically, there is a local decondensation, and these puffs are the sites of active transcriptions. There are beautiful studies, classical studies, where tritiated uridine has been used, and then it has been shown that all the uridine, the radioactivity is found only in the puffs and not in other regions. Okay. Then there are other studies using the immunofluorescence, where you have antibodies tagged with the RNA polymerase 2, and then it is conjugated with the fluorochrome, and you can see the fluorescence, they coming up in only the, so RNA pol 2 can fluoresce only on those regions where it is transcribing, not in other regions, and so on. So this is the heat shock puffs, 87A, 87C locus. Like after the flies are transferred to transferred to the higher ambient temperature. So these are the these are the two regions of the polyethylene chromosome puffed out, and they transcribes, and they are the sites, the loci for HSP70, heat shock protein 70. So these are the studies where you can use a polyethylene chromosome, and quickly you can find out the potential. Now I will end just citing some of the published work on Drosophila from my laboratory. Quickly, I'll give a glimpse. So these are one of the very important piece of work we have published, and it is actually doctoral work of Lina Thorat. She is Dr. Lina Thorat, and then we first showed that the trehalose, which is the major sugar in the insects, it level goes up. And this can be an indicator of the desiccation stress in Drosophila. By the way, Drosophila, the name originates from the two words. Dros means the dew, and phila means loving. So dew loving, that is a humidity loving flies. So when such flies are exposed to conditions where the availability of the water or the atmospheric and the humidity is less, so it's just when it is stressed under desiccation, then we have seen the trehalose level goes up. And then it can be an indication of the anhydrobiosis. That is, anhydrobiosis word means the life without water. And this is a publication first in the BBRC, and then it is followed by other publications. Like it has then it was again Dr. Lina Thorat's doctoral work, and we have shown that there is a change in the life history traits. There's a heterochrony. Then the ecdysone, that is a molting hormone, there's a shift. And this work featured in the cover page of the Journal of Bioscience. You can see this is desiccated larva. under extreme desiccation stress, but again, it has a remarkable ability to revive within a certain, the time period, and then it can revive when the function well. So this remarkable ability of Drosophila flies to adapt to stress is we are currently working on it, and there are other papers. This is a collaboration with Dr. Shubhra Chatterjee in Anna University, and and then there is a collaboration with Dr. Um, uh, Alexander Breming of Marburg University, where the Dr. Lena generated flies, which is like is a knockdown of a TPS. And then if you look at this pathway, there's a trehalose phosphosynthesis, the first step in the trehalose biosynthesis. And now if you downregulate, that means these flies are deficient of making trehalose. Even under, so basically under desiccation stress, the trehalose level goes up because this pathway is active. Now, if you just knock down TPS, then what happens? So this was actually a very interesting paper from our side, and then where we can show that, okay, basically this is the wild type flies, and the desiccated fly, they have a desiccation tolerance threshold is very low. But if you feed them with the external trehalose, that is supply trehalose, which is they are not being able to synthesize, but if you supply trehalose through feeding, again there's an improvement. So, and the other live imaging we have shown in this paper. Then Pratibha is just will be submitting soon. Pratibha, she is working on an interesting aspect that what happens when there's a multiple stress, administered to Drosophila. So that means, like basically what happened, Drosophila and also in Carinomus. Like, they generally, most of the studies, people work with one stress at a time. But in the natural condition, multiple stress operate on a single individual. So we are addressing such a complex, the question, this is a Pratibha's main focus of the doctoral work, where the combination of different stress, more than one, and within singularly or plurally, what happens. And so this is the first the paper published in European Journal of Environmental Science, 2019, and the other paper are in the pipeline and under review. There's another, the collaboration work with Dr. P.K. Khanna, who is a nanobiotechnologist, and we started working on the nanogenotoxicology. This is Dr. Kanchan Pathak. She just got her degree recently. She published the first paper in 2016 that Drosophila melanogaster, if you administer or expose them, feed them with the silver nanoparticle and also the macroparticle, it affects the pigmentation pathway, and which is called leucism. And the leucistic phenomena, you can see the total loss of the pigment of the flies, except the eye. So eye pigment is a totally different pathway, but the other pigmentation pathway, which is actually the the tyrosinase, phenol oxidase, DOPA, and melanin, that gets perturbed, and you can see that the total absence of the pigment. And the other implications of these studies, if you read the paper, you can get enlightened. Now, I'll come to the last slide, the future directions. And so I would like to end by saying, by giving a message that many interesting biological processes and avenues remain to be investigated in Drosophila. Many challenging questions can be answered very fast to explore biological phenomena which remain unanswered till date. Moreover, Drosophila has proven to be an efficient translational model system for learning and researching in the areas of biomedical and environmental interdisciplinary sciences. So they're the people who has shaped my, scientific quest, which I tried to address many times in Drosophila and Carinomus. Many of them worked with the Drosophila also, like this. And in my lab, most of the people, that they work both on Drosophila and the Carinomus, because I use the Carinomus to find out evolutionary answer, because the Carinomus, the flies, Carinomus, the midges, they evolved. There's a different flies. in 200 or 250 million years ago. Drosophila evolved 100 million years later. So many of the things which we find in Carinomas, we do not find in Drosophila. So this type of ecological and like evolutionary questions we keep on asking. Okay, and these are the new group of the students like Prachi, Sneha, Snehal, Amreen. They're working. Kanchan they got a PhD degree. She worked on Drosophila and nanotechnology. Chetali, Minal, they're working. Pratibha is submitting, and the other people they've contributed a lot. So thank you very much, and thank you for your patience. Hello, Dr. Kale. Usha, madam.
Yes, sir. Praveen sir, are you reading the questions?
Sir, can I ask?
Yes, sure.
Sir, I am very, there are two questions, one related to this work, another one, you showed the deletion and all that. How did you use the video?
This is not a video, this is we made it.
You made it? Very nice. How did you make that?
Oh, there are lots of tools are available. You can ask your students. They're very
okay,
smart.
How they cut, and then because it was very beautiful. And another thing, what did you use for inducing? Sir, you showed very beautiful. It was just like book. The type of when you have deletion and in that packaging stage, the way you showed the looping.
Oh no,
beautiful.
This is okay. So let me just tell you, you need a very good
microscope.
Microscope, more than microscope, the very good finger. The hand is important.
Yes.
So out of experience, but the protocol which is many time it is followed. It is not suitable for research. For classroom, practical, UG and PG, it is okay. Just to show that yes, there is a polygyny chromosome.
Yeah, no, I'm talking only about UG PG, sir.
Oh, okay.
Suppose UG PG, I wanted to show this. Okay. Then what type of this I should use?
Okay, I just send you the protocol. I just send you the protocol.
Fine.
And
no,
also
only UGPG. We are not.
But DBT Star College protocol, which DBT has released. There's
that
a
book. I have the book.
I have a chapter there. I don't know whether you go to the.
Fine. I'll go through it again, and we will standardize it.
Page number 56 to 69. That's my chapter in the DBT Star College. Yeah.
Fine, because very beautiful and very nice. We also get some very good preparations.
Yeah, but let me just,
as I said, this is one of our favorite materials.
I just tell you, I just give an analogy. Like when you cook something, you buy masala, the spices for ready-made, or sometimes you make it. There is a difference. So yes. I prefer to make stains myself. I do not buy ready-made this stain.
So we also don't buy.
But again,
don't buy any ready-made stains.
But this recipe, making the protocol is very, very important.
Recipe.
And the staining protocol, like you have to age the stain, and then there are when you dissect the glands, there should not be any other non-glandular tissue, because that disturbs the cytological clarity.
Correct.
Okay, and that there are different type of clearing medium. Like the many people use after they just put the stain and then they squash on that stain itself. But there are many the clearing media is there, like propionic acid, then like the acetic acid, glacial acetic acid, which is the low percentage. So this helps to soften the tissue and yet retain the stain, so it doesn't distain. I can just give you all the audience that many of the classical the beautiful pictures we see in the textbook of a chromosome picture, it is even the mammalian chromosomes, it is done with the 1:1 is a mixture of orcine and carmine. You try that, it's very good. So you have to make yourself like orcine carmine combination 1:1.
Yeah, because orcine is better than carmine. In carmine there are sometimes too many crystals, even if you try to
so basically
filter it or centrifuge it.
So you can use a combination, or like I said, 1:1 ratio.
Okay, 1:1.
But it has to be fresh at the time of use. Yeah, so you have a working solution, standing solution, you make it a fresh one and then apply.
And I also like that experiment on desiccation which you showed.
Yeah.
And so you measure the amount of Trellos which is formed around.
Yeah.
Okay.
Actually, you
so
can.
That's an indicator that.
Okay. In the college level, you can just use a spectral method with the Anthron test. But they sometimes they if you want to publish, they demand HPLC grade. But Anthron test is fine. Okay, Dr. Kale, sir, you have asked, have you identified the chaperone protein formed in response to stress and the role in stress tolerance? Yes, it is published. We have done this in the Carinomus, not in Drosophila. It is unpublished here. So we have done that.
Okay.
And other questions?
Yeah, sir. Next, yeah. Sangeeta madam.
Yeah, what protocol is employed for multiple stress factors on the fly? This paper I can share where there is published already, and unpublished ones, I'll just retain till Pratibha submits her thesis. But the protocol for employment of the multiple stress, European Journal of European Journal of Environmental Science is freely downloadable. I'll just send you the. You just type Nath, Bomble, and Nath. E.g. European Journal of Environmental Stress 2019, you can freely download, or I can send it. Then Dr. Kalisar has another question.
Yes.
Are the micronuclei developed in Drosophila in response to exposure to stress condition? Which tools are used in following the cytogenetic changes in Drosophila? Excellent question, Kalisar. Such type of question comes from you only. Yeah. but the Drosophila, if you use the polyethylene nuclei, there is no division. So when there is no division, you do not get micronuclei. The answer number one. If you want to see the micronuclei, then you have to use the hemocytes, which are floating in the hemolymph. So hemocytes, you can just isolate from the hemolymph, okay, enrich them, and then they divide actually, and then you can find out micronuclei. I have not done it, but the that can be done. But the problem with the insect hemolymph and the putting them into division cycle, it's quite cumbersome because lots of like debris come and so on. But polyethylene nuclei, you cannot see this. You can show this because micronuclei, but it's one of the tests, as you rightly said, for find out genotoxicity. But that cannot be done. Okay, then another.
Yeah, another question from Kale sir only. Yeah, have you identified the chaperone protein?
Oh, this I've answered. There's another question in the beginning. It's a long question. This is not for the public. So Sangeeta madam, you can just send me a mail to bbnath@gmail.com and we can discuss these issues.
Yeah.
Please. And I think I do not find many questions. And these are very encouraging feedback, and that's my reward.
Yeah.
But many times, in the chat box, and people do not write negative comments. So even if it is bad, so
there's no negative, sir.
No. They can, I just request the audience that they can just send their feedback so that I can know the ground level like reactions, because the generation change, we also evolve as a teacher.
Yeah.
I hope the popular lecture series I could retain the content at the popular level, not going deeper into very specific tech. I have avoided the technique-oriented talk.
Praveen sir, do the needful.
Yeah, I was seeing the thank you. What is this? So like a classroom, Drosophila classroom. The teacher and the students are all Drosophila. Okay, sir. Yeah, should I propose a vote of thanks, sir?
Yes, yes, please.
Okay. On behalf of Association of Teachers in Biological Sciences, ATBS, and the Department of Biological Sciences, I extend our gratitude and thank Dr. Bimalendu Nath, sir, for sparing his valuable time and giving an excellent talk. The animations, what has been used, were also excellent, and we learned a lot. So on behalf of ATBS and Jhunjhunwala College, I thank you, sir. Also, I thank the members of the ATBS as well as the teachers from different colleges and our college, student of our college and different colleges. I feel there are a few Nath sir students also who have been attending. So on behalf of ATBS and Jhunjhunwala College, I thank all these members for having attended this webinar and listening to the talk given by Nath sir. I also express my thanks to the technical assistance given by Mr. Devi Prasad Shetty. Thank you once and all.
Thank you very much, Nath sir. But one request.
Yes, yes.
If there is some good student, do you think you can give 1 or 2 days training?
Yeah, yes, yes. No problem. Definitely.
Not for research, sir. They are basically
No. Okay.
protocol.
No problem.
Thank you, sir. Thank you. So wonderful. I'm very impressed with that. And even that mitosis.
Thank you.
बहुत ही सुंदर था.